Hello guys, so now we will start with the third episode of the systemic histology. In this we will cover hepatobiliary system and pancreas. First we will start with the hepatobiliary system. It includes liver, gallbladder and extrahepatic ducts. So we will start first with the basics which will be asked in the viva. So it is, as we can see, it is a liver. It has a larger right lobe and a smaller left lobe. The liver is present in the right hypochondrium region of the abdomen. We have a hepatic duct which is right and left hepatic duct which drains from the respective sides of the liver. Both the right and left hepatic ducts combine to form a common hepatic duct, and as we can see here, it is a pouch sac like structure which is nothing but a gallbladder. It has three parts: fundus, body and a neck, and then it forms a duct which is called as a cystic duct. Cystic duct combines with common hepatic duct to form common bile duct. Common bile duct, it combines with the duct of the pancreas, major duct, which is also called as a duct of Wirsung. It joins to form a hepatopancreatic duct which opens at the major duodenal papillae of the duodenum. So now we will see pancreas. Pancreas, it has four parts. It is a head, neck, body and a tail. As we can see, there is a duct arising from the tail and it divides into two parts. The one which opens at the lower end is called a main pancreatic duct, from which most of the secretions are secreted, or it is also called as duct of Wirsung, and a minor duct which opens at the minor duodenal papillae of the duodenum, which is called as duct of Santorini or the accessory pancreatic duct. And we can see here it is a common bile duct which is combining with the major duct and opens at the major duodenal papillae. And we can see here we have a many capsules which is dividing the pancreas parenchyma into different lobes. Now we will start with the histology of the liver. First we will study its basic. So it is the largest gland, not a largest organ. Largest organ is the skin. It is the largest gland of the body and has both endocrine and exocrine part. Even pancreas has a both endocrine and exocrine part. It is situated in the right hypochondrium and it weighs about 1.5 kg. It has a dual blood supply. It receives two blood supplies. One is portal vein and other is hepatic artery. And it is the accessory gland of the GIT. For example, salivary glands, liver, pancreas, these all are accessory glands of the GIT. The endocrine part secretes plasma protein, as we have seen albumin, globulin, prothrombin, clotting factors are secreted by liver, and exocrine part it secretes bile. The liver has many functions in our body, such as metabolism of the carbohydrate, proteins and fats. It synthesizes the plasma protein, fibrinogen, prothrombin, clotting factors, etc. It regulates the blood glucose level and lipid levels. It helps in the storage of the glucose as a glycogen and also stores vitamin A. It helps in the degradation of the drugs and alcohols so they can be reduced to the less toxic form. It removes bile pigments. It is the center of RBC production in the fetal life. In the adults, it is not the RBC production site. In the adults, the bone marrow is the RBC production site. And it has a Kupffer cells which are nothing but a macrophage, so helps in the phagocytosis. Now we will see how a parenchyma of the liver is divided. So parenchyma of liver is divided into three different lobules, but mainly which is important is hepatic lobule, which we will study in detail. So now we will compare all the three lobules: hepatic lobules, portal lobule, portal acinus. So as we can see, it is a hexagonal shaped. In the center we have here central vein, and at vertices it has three structures. It is nothing but a portal triad. So it has a total of 6 portal triads and it is a hexagonal shaped. Portal lobule. So it is a triangular in shape. As we can see, it forms by joining 3 central veins. Next we will see portal acinus. It is a diamond shaped. As we can see, it is a diamond shaped and it forms by joining 2 hepatic artery and two central veins. Now we will study in detail about the hepatic lobule, which we will see under the microscope later. Hepatic lobule, it is a hexagonal shaped and has a central vein at the center and portal triad at the vertices. So 6 vertices, 6 portal triads. Portal triad contains 3 structures: portal vein, hepatic artery and bile duct. There are hepatocytes, which are nothing but cells of the liver. Hepatocytes are arranged in a plates or a lamina, which are separated from the each other by the sinusoid, which is a blood-filled space. As we can see here, these are the hepatocytes, which are arranged in a single row or a plate, and between the hepatocytes there is a red colored space, which is nothing but a sinusoid. It is filled with the blood. And we can see center, there is a central vein, and at the vertices we have a portal triad that contains hepatic artery, portal vein and a bile duct. Sinusoid is lined by the fenestrated endothelium and Kupffer cells and a natural killer cells. Now, as we can see under the histological diagram, it has a central vein and the hepatocytes are arranged in the plates, and in between that we have a sinusoids, and at the side we have a portal triad. Under the microscope, as we can identify, there is a central vein, and just little distance away from the central vein we have a structures which appear as a dilated veins and arteries. These are nothing but a portal triad. As we can see here, it is a portal triad. It is also a portal triad. We can appreciate it, and we can see here there is a hepatocytes which are arranged in a plates, and around it we have a white space, but which is nothing but a sinusoids. So how we will identify liver? To identify liver, we should identify first a central vein, which will appear as a clear and sometimes may have a red dots within it, which are nothing but RBCs. And hepatocytes are appear as a pink colored, and a white light area is nothing but a sinusoids. And portal triad will be seen just little away from the central vein. Now we will see histology of the gallbladder. It is a blind muscular sac. It contains bile, which is secreted from the liver. It helps in the concentration of the bile and remove it by the cystic duct. It does not have muscularis mucosae and submucosa. As we have seen, the GIT has a 4 layers: mucosa, submucosa, muscularis externa and serosa. So in this, the part of mucosa, that is muscularis mucosae and submucosa, is absent, has only 3 layers. It has mucosa, fibromuscular layer and serosa. Fibromuscular layer, it is nothing but a muscularis externa layer, and a submucosa, as I said, submucosa is mainly made up of a connective tissue, so it is a fibromuscular layer. It's nothing but submucosa and muscularis externa are combined into form a single layer called fibromuscular layer. I have gave a trick to remember the epithelium. From the stomach till the upper part of the anus is lined by simple columnar epithelium. So it also comes in between that. So it is also lined with the simple columnar epithelium. And the lamina propria is thrown into a numerous folds. As we have seen, esophagus is the only part which does not have folds. Rest everything has many folds, but it does not contain any villi. Fibromuscular layer, it has muscularis externa layer, and but it is has a less muscle content and has more fibrous content. And serosa is made up of the connective tissue. As now we can see here, it is the lining epithelium, which is made up of a simple columnar epithelium, below which we have lamina propria. And we cannot see any muscularis layer, muscularis mucosa layer in between, and no submucosa. Directly we have a smooth muscle layer, which is also made up with a fibrous tissue. So these both constitute the fibromuscular layer, and below this we have a serosa. Now under the microscope we can identify easily. We have a many irregular folds and columnar cells, and we can see there is a fibrous layer, and directly we have a little muscle layer and mostly made up of a connective tissue. So it is a fibromuscular layer, and below this we have a serosa. How we will identify it? By looking the irregular mucosal folds, we will have a less smooth muscle content, that means a less dark pink colored area. Many different colored layers will not be seen, only 3 layers will be seen. As we can see here, it is the epithelium, it is the lamina propria and it is the muscle layer. Only 3 different layers will be seen. Histology of the pancreas. Pancreas, it is the retroperitoneal organ and extends from the C curve of the duodenum till the hilum of the spleen. So it is the duodenum. From here it extends till the hilum of the spleen. It is the hilum of the spleen. It is the C shape of the duodenum. It has four parts, as we have seen. Pancreas, it is the head part, it is the neck, it is the body, and it is the tail. And it has both endocrine and exocrine part. Exocrine part secretes watery secretions, mainly contains serosacinae. So it secretes watery secretion and certain enzymes for the digestion, trypsin, chymotrypsin, lipase, etc. So what is meant by exocrine? Exocrine glands, these have a duct. So as we have seen in the pancreas, we have two ducts, major and minor pancreatic ducts. So that is the exocrine part. And endocrine part, it does not have any ducts and it secretes hormones. So it has three types of cells which are also called as islets of Langerhans. So it is important to remember because it will be asked in the viva. We have alpha cells, beta cells, delta cells. Alpha cells secrete glucagon, beta cells secrete insulin, delta cells secrete somatostatin and gastrin. What is function of glucagon? Glucagon helps in increasing the blood glucose level, whereas insulin is antagonist of the glucagon. It decreases the blood glucose level. And delta cells secrete somatostatin and gastrin. Somatostatin inhibits the secretion of the gastrin, which is required for the HCl secretion, and gastrin promotes the secretion of the HCl. It helps in the most of the digestion and regulation of the blood glucose. As we have seen, the intestine, small intestine, is the major site of digestion and absorption. It has a capsule which divides parenchyma into lobules. Liver does not have capsule which divides into lobe, but normally liver has a capsule surrounding it, which is called as a Glissen's capsule. But in the pancreas, we have a capsule which divides parenchyma into lobes. So as now we can see here, it is a pink color which is a capsule, and around it we have a serous acini because pancreas is mainly made up of a serous acines. So these are pink in color, and as we can see here, it is cells which are arranged in a groups which are appearing as a white color. These are nothing but islet of Langerhans, which secretes insulin, glucagon, somatostatin, gastrin, etc. And now we will see under the microscope. As we can see here, it is also appearing as a pink colored as we have seen in the liver, and it has white spaces around it, same as the sinusoids which are present in the liver. So how we will identify a pancreas? There is no central veins are seen, and we have a larger white zone which are nothing but islet of Langerhans. It is the main identification point of the pancreas, and we can identify here we have a septa, which is nothing but a fibrous capsule. So I have said fibrous capsule divides a parenchyma into lobes in the pancreas and does not divide into lobes in the liver. It is the main identification point. Islets of Langerhans and fibrous septa divides into lobes. So identification point, serous acini will be seen as a complete pink color like liver, and the light circles will be seen as they are the islet of Langerhans. It is the most important identification point, and fibrous septa will be seen. And here we complete our third episode. Now in the next we will start with the urinary system. For the notes you can visit my Telegram channel. I will provide link in the description. Thanks for watching. If you love the content, please like, share and subscribe.